The central dogma of biology. I always feel like there should be a fanfare playing when I say the central dogma of biology. Central dogma of biology is really just an explanation of how proteins are made using the information that we find in DNA. So in the last lecture we talked about DNA and DNA replication. This time we're going to talk about how we use that information in order to synthesize proteins. And it's really two steps. The first step here, transcription, and then translation. Transcription is when an enzyme called RNA polymerase reads the DNA and synthesizes a copy. And the copy is made in RNA, a very similar molecule to DNA. And then translation is when the RNA information is used to actually synthesize a protein or a polypeptide. Now the terminology here, transcription and then translation, the reason they named it this way, transcription, like when you transcribe or copy something, is going from the language of DNA into the language of RNA, and those are almost identical molecules, and it's nucleotide for nucleotide copying. When you go from RNA to protein, however, you are moving from nucleic acid information in RNA into amino acid information in protein. Totally different. You're translating between languages. That's why they named them that way. If that doesn't help you to remember it that way, they also go in alphabetical order. So transcription and then translation. Now, the flow of genetic information here. Of course, all information is originally in DNA inside of the cell. And the DNA goes through replication on the left-hand side of this picture that we talked about before, through reproduction in order to give rise to new organisms. The other thing that the DNA can be used for, of course, is to make the proteins through transcription and then translation. Now in transcription we know that we have copying of the DNA message, and that copy then is made into RNA, several different kinds of RNA here. While we're here, let's say a couple of words about RNA. RNA is ribonucleic acid, very similar to DNA, which is deoxyribonucleic acid. Same basic structure. It is made of nucleotides, RNA is. It tends to be single-stranded, however, instead of double-stranded. Almost always it's single-stranded. There are several different kinds of RNA that you're seeing here. mRNA, which is messenger RNA, transfer RNAs, ribosomal RNA, several different kinds of RNAs there. So it has ribose for its sugar instead of deoxyribose, and the other real difference here is that RNA does not have T or thymine as a nitrogenous base. Instead, it has the nitrogenous base uracil, U. So everywhere that you would find a T in DNA, you're going to find the U in RNA. Alright, so here's the different kinds of RNA that are made during transcription. The main one we think about here is messenger RNA. That's the direct copy from the DNA that's made during transcription. tRNAs are going to be involved in the process of translation. rRNAs make up the part of the structure of the ribosome. After transcription, then all of that RNA, it goes through translation, and translation, the ribosome is going to use the mRNA as well as tRNAs to actually make that protein. We'll go through these one step at a time. To start out, let's start out with transcription here and look at this process. Transcription is the reading of the DNA again. It's synthesizing an RNA copy, and it synthesizes a complementary RNA copy from the DNA. The enzyme that does this is called RNA polymerase. So RNA polymerase is going to read the DNA and synthesize a complementary mRNA copy. And so it's in white here, reading the DNA and synthesizing a complementary mRNA copy. Interestingly, it goes in the same orientation as DNA polymerases did. Notice it's making new RNA 5' end first, 3' end last. Now, how does this work? RNA polymerase is literally reading the bottom strand of the DNA here And as it reads a nucleotide on the strand, it's putting in the complementary one. So if it reads a C on the template strand here, it puts a G into the growing one. If it reads a T in the template strand, it puts an A on the RNA. If it sees a G, it puts a C on the RNA. If it sees an A in the DNA, it puts a U into the RNA. Remember that the base pair rules here are C to G and A to U for RNA. All right, so RNA polymerase is the enzyme doing this job. It starts at a particular sequence in the DNA we call the promoter. That's a very specific sequence in the DNA where RNA polymerase will bind to start transcription. And it will go through that process until it reaches another sequence we call the termination sequence, then it will literally fall off the DNA. So RNA polymerase starts at the promoter and ends at this termination sequence. Here's another picture showing transcription. This is one from your textbook. It's a little bit more complex. The enzyme here is RNA polymerase. You see it opening up the double-stranded helix, binding to the promoter, and then it begins to synthesize mRNA, a complementary anti-parallel copy. And notice it's doing 5' end first, 3' end last. And how does it know which nucleotide to add? It adds nucleotides according to the nucleotides it reads in the template strand. Right? It sees a C there, puts a G into the RNA, for example. This process will continue. RNA polymerase will continue to copy until it reaches a sequence in the DNA we call the termination sequence. All right, a couple of terms here that are used commonly with transcription. The term downstream versus upstream. Downstream is the direction of transcription. Upstream is the opposite direction. So downstream is the direction of transcription, while upstream is the opposite. So for example, if you think about RNA polymerase as being sort of something floating on a river, the direction it's moving is downstream, right? That would be downstream. The other direction would be upstream. So how you could use these terms. The termination sequence is downstream from the promoter. The gene that's being transcribed is upstream of the termination sequence. Right? You could use the terms that way to indicate a directionality. So kind of a recap of transcription here. In transcription, RNA polymerase binds to the promoter, which is upstream of the gene that's being transcribed. RNA polymerase then adds nucleotides complementary to the template strand, and it does it in the 5' to 3' direction, and it's building mRNA. Remember that RNA never has thymine in it, so instead uracil is used wherever you would expect to find thymine. So the base pair rules for RNA are C to G and A to U. RNA polymerase falls off the DNA and stops transcription at the termination signal when it finds it. Now, I want to pause here and mention that I do have a video that I posted in the same week's folder on transcription. I found it on YouTube, so if you'd like, I would suggest going in and watching that video. It's kind of a recap of what we just talked about, and it's nice to see it in video form. I don't want to do it while I'm lecturing because I think that that makes very large lecture files, which tends to be a problem in the playback, so that's why I'm not doing it right now. All right. So I want to orient, where are we again in this whole system? We have our double-stranded DNA here. We've just talked about transcription, which is making the mRNA, the messenger RNA, and that of course starts at the promoter, ends at the termination sequence, and utilizes an enzyme called RNA polymerase. Interestingly, RNA polymerase doesn't need all of the assistance that DNA polymerase did. Remember when we talked about DNA replication, it's a much more intense process with lots of different enzymes going on. RNA polymerase actually unwinds the DNA on its own during transcription, so it doesn't need additional help. All right. Let's talk about translation now, using the information that we just produced, this mRNA copy, in order to produce a protein. Now, translation. Before we get into how that works, I want to mention how the message is even going to be read. This is a diagram that's showing DNA, mRNA, and then protein. In pink here, they're showing the double-stranded DNA helix. You could label this 5 and 3 and all that to make it anti-parallel. They're trying to show you that it's complementary base paired through shapes, right? That's what they're indicating. So, if you look at this, first off is transcription. Transcription happens here in between the red and the yellow. Transcription is happening there. Transcription is done by RNA polymerase, and you wind up with this yellow copy from the DNA, and that is the mRNA, the messenger RNA that was produced during transcription. Now, that's a direct copy, nucleotide for nucleotide. RNA polymerase reads this nucleotide, and it synthesizes a complementary antiparallel copy. Then it read this nucleotide, and it synthesized this one. So it is nucleotide for nucleotide, right? Each one is read, and then the complementary nucleotide is produced. In translation, however, it's not read one for one. In translation, the mRNA is read in 3 nucleotide chunks we call codons. A codon is 3 nucleotides that encode 1 amino acid. I'll say it again. A codon is 3 nucleotides in the RNA that encode 1 amino acid. So during translation, when we're moving from the message of nucleic acid into the message of amino acid or proteins, what's going to happen is this mRNA is read these 3 together, and that's the information for the first amino acid. Then the next 3 are read, and that's the information for the next amino acid. And then the next 3 are read, and that's the information for the third amino acid. So it's read in codons like that during translation. The actual molecules that do that process are called tRNAs, or transfer RNAs. Transfer RNAs are going to interpret the DNA code and actually translate the information from DNA that was then into RNA and transfer that into amino acid language. So let's look at this picture for a moment. Here we have our DNA, right? Double-stranded. You could make it anti-parallel, putting in 5s and 3s there. It is complementary base paired. They're showing you A's to T's, C's to G's. It is complementary base paired. This first set of arrows, right? In the first set of arrows, transcription is occurring. RNA polymerase bound to the promoter. It started transcription. It's copying nucleotide for nucleotide and actually making the complementary one. As you look at this, notice, here's your pink mRNA. Which strand of the DNA was transcribed? Look through and identify which strand of the DNA was transcribed. The top strand, the red strand, was transcribed. How do I know? Because RNA polymerase synthesizes a complementary copy. So it read the T here and it put in the complementary nucleotide, which is an A. It read the A here, it put in the complementary nucleotide, which is a U. It read a C, put in a G, for etc. down the line. So that pink mRNA is a transcript from the red template strand. Okay. Now, what's going to happen in translation is molecules called tRNAs or transfer RNAs are going to read that codon, the 3 nucleotides, and bring in the correct amino acid. And it's the transfer RNAs that actually do that job, reading the mRNA and bringing in the correct amino acid. Here's how it works. Here's the structure of a tRNA. You can see why they call it T. It looks very much like a T. At one end of this RNA molecule is what's called the anticodon. Notice this anticodon, get my arrow back here, this anticodon will bind through complementary base pairs to the codon of the mRNA. So what you're going to see is the anticodon binding to the codon, and this tRNA also brings in an amino acid. And that's how the translation actually occurs. Then the next tRNA has a different anticodon. He's going to bind to the next codon through complementary base pair rules and bring in the next amino acid, a different one. Different tRNAs have different anticodons, and they bind to different amino acids. And that's how the translation actually occurs, is the tRNA that has the correct anticodon to bind to the codon of the mRNA is going to bring in an amino acid. And that's what does this translation into the message, into the language of protein and synthesizing this protein. We're going to go through this in more detail, but that's sort of the big picture of how this works. All right. Also, I want to mention, here's the actual code. How do we know what protein to make, or how do the tRNAs decide what protein to make? Well, remember, it's all about the codon that's in the mRNA and the anticodon that's in the tRNA. What you're looking at here, this is the actual chart for how a protein is produced. In other words, which codons in the mRNA encode which amino acids. Right? So for example, if the mRNA has a codon that is CCA, it's going to encode the amino acid proline. If it has AGU, it's going to encode the amino acid serine. Right? So this is the actual machinery that to change the message from one language to the next. Right? While we're here, I do not want you to memorize this code. I don't want you to memorize exactly which codons encode which amino acids. You don't need to do that. But I do want to point out a couple of things about this code. First off, notice there is a start codon, and that is AUG. Translation will always start at an AUG. That is the start codon. It turns out that AUG also encodes the amino acid methionine. So for that reason, all proteins have methionine as their first amino acid. All of them do, because methionine is always encoded by AUG. Likewise, notice we have some stop codons, UAA, UAG, UGA. Those are the three stop codons. That's where translation will stop as the process is going. Once those codons show up, that's going to force translation to stop. Right? So I do expect you to know that AUG is start, UAA, UAG, and UGA are stop. The other thing I want to point out, there is what's called redundancy in the code. In other words, there are multiple codons that encode the same amino acid. For example, if you have GCU, you're going to get the amino acid alanine. But if you have GCC, you're going to get the amino acid alanine. And if you have GCA or GCG, you're going to get the amino acid alanine. Right? That's because of this redundancy. Multiple codons encode the same amino acid. The other thing to point out here, this code is universal, which means all organisms on the planet use the same code. So CUU in a human mRNA encodes the amino acid leucine. CUU in a frog's mRNA encodes the amino acid leucine. CUU in bacterial mRNA encodes the amino acid leucine. And this is why you actually can take DNA from one organism, put it into a different organism, and you'll get the correct protein being made. Because this message, this code, is universal. So it does actually allow the production of the correct protein in different organisms. All right, the site for all of this activity for translation, of course, is the ribosome. You already know quite a bit about the ribosome. Remember, the ribosome has two subunits, the large subunit and the small subunit. The ribosome is made of both protein as well as ribosomal or rRNA. That is more important than you might think. It appears that the RNA of the ribosome is some of the most important portion of the structure and function. So yes, the ribosome is not just protein, it is also made partially of rRNA. All right, another thing to notice, the ribosome has two main sites for binding of tRNAs. They call them the A and the P sites. The E site doesn't really exist. It's sort of an exit tunnel for tRNAs. You'll see what I mean in just a moment. Okay. So two subunits. The subunits are made up of both protein and rRNA. And there are two binding sites for tRNAs in that ribosome structure. Notice when the ribosome is actually functioning and active, the mRNA is sort of between the two subunits here. And the tRNAs, look at how they're arranged here. The tRNAs, here's his anticodon. Right? His anticodon can bind to the codon of the mRNA. And this one, his anticodon, can bind to the codon of the mRNA. And they're bringing in different amino acids. All right, let's look at how this process goes. First thing that happens, it actually happens before we're seeing this slide, but first thing that happens, the small subunit binds to the mRNA, and once it finds that start codon AUG, then the large subunit attaches. All right, so at this point, we have our mRNA, and we have the ribosome assembling around the mRNA. Now, the first tRNA is going to come in and bind here to this P site of the ribosome, and his anticodon is binding to the codon, the start codon of our mRNA. And of course, he's bringing in the first amino acid, and you know the first amino acid is always methionine, because methionine is what's attached to the tRNA that binds to the start codon. All right, so once again, ribosome assembles around the mRNA, and it scans looking for the start codon. Once it finds the start codon, AUG, in comes the first tRNA to the P site, and its anticodon binds to the codon. It's bringing in the first amino acid. The second codon of the mRNA, right, lines up with the A site. The second tRNA comes in and he binds his anticodon to the codon of the mRNA, and they're binding through complementary base pair rules. The second tRNA is bringing in his amino acid, which happens to be leucine. A little protein bond forms between the two amino acids, right, to attach them together. Now, the ribosome begins to shift. It's pulling the mRNA through. Right? That forces our first tRNA through the exit tunnel and out. It's going to move this tRNA into the P site, and notice the two amino acids are now connected to one another. Right? Next, the third codon is now lining up with the A site, so the third tRNA can come in. Once the third tRNA is in there, his anticodon is binding to the codon of the mRNA. And of course, he brought in his third amino acid. Little peptide bond is forming between these two. Right? The mRNA is moved once more through the ribosome. Right? Second tRNA is kicked out through the exit tunnel. Now we have this third one in our P site, the three amino acids connected together. Now the fourth tRNA can enter into the A site. He's going to bind to the codon of the mRNA through his anticodon, bringing in the fourth amino acid. Peptide bond forms between them, and then he's going to pull the mRNA through, kicking out the old. So the basic process here is the ribosome assembles around the mRNA, scans for that start codon, and then the tRNAs enter into the ribosome sites one at a time, binding their anticodon to the codon of the mRNA. They bring in an amino acid. The amino acids attach together to form the protein. And then the ribosome moves the mRNA through, kicks out the old tRNA, allows the new tRNA in. Right? And step by step, that's how a protein is built. I'm going to go back through it. All right, so starting here, we have our two subunits assembling together along the mRNA. The start codon is where everything starts here. At the start codon, the first tRNA is entering. His anticodon binds to the codon, and he's bringing in the first amino acid, which is always methionine. At the A site, our second tRNA binds his anticodon to the codon, and he's bringing in his second amino acid. Peptide bond forms between the two, and now what's going to happen is the mRNA is going to be pulled through. Right? mRNA is pulled. You can see it showing in this direction. That's kicking out the first tRNA through that exit tunnel. Second tRNA moves now into the P site. Now we have the next codon available. So the next tRNA enters. He's going to bind through his anticodon to that third codon, bringing in the third amino acid. Peptide bond forms between them. Now the mRNA is moved one more notch down. Old tRNA gets kicked out. Amino acids are connected to the tRNA in the P site. Now the fourth one can come in because we have a fourth codon available. He's going to bind via his anticodon, bringing in the fourth amino acid. Peptide bond forms between them. mRNA will move, and once again kicking out the old, bringing in the new. This will continue to go until the ribosome encounters the stop codon. When the ribosome encounters the stop codon, literally everything flies apart. Once it hits the stop codon, the two subunits separate out. The protein that was formed is released. The mRNA can be used over and over again, so you can get multiple production of proteins from one piece of mRNA. At this point, I want to mention that I also am posting a video for translation. So if you want to see translation in action, which is really a cool video, you should check that out. It's also in the same folder there. What is that, week 6 folder, I believe? Yes, week 6 folder. That's posted and available if you'd like to see that. I also have another video that has transcription and translation together. So there's, I think, a total of 3 there that you can look at for transcription and translation. All right. So I mentioned this, and I want to mention it again. One piece of mRNA can be used for multiple different ribosomes. So you can get lots of protein being produced from one piece of mRNA. That allows lots of protein production from just one piece of mRNA. In fact, in a prokaryote, you can even have translation starting before transcription is done. So let me point out here what's happening. We have our double-stranded DNA. Right? We have RNA polymerase opening up the double-stranded DNA, copying the DNA, making mRNA in orange here. Okay? He started at the promoter. He's going until he hits the termination sequence. He's synthesizing a complementary anti-parallel copy of mRNA. Now, before he's even done, before he's even hit the termination sequence, already the ribosome is assembled and has found the start codon. And the ribosome begins to use tRNAs to produce a protein as it reads that piece of mRNA. And you can have multiple ribosomes doing that. Right? So you can get protein being produced even before transcription is done. That allows very fast protein production in prokaryotes because they're so quickly making protein even before transcription is done. What you're seeing here, it looks like beads on a string. This is this polyribosomal complex. This is one long piece of mRNA and multiple ribosomes on it. And each of those ribosomes you can see are synthesizing a protein. Right, so here you can see the protein that's being made by that ribosome. Lots of ribosomes along one piece of mRNA. Now let's talk about some of the differences in transcription and translation between prokaryotes and eukaryotes. I just talked about how in a prokaryote, transcription and translation can occur at the same time. Right? So is that true for a eukaryote? Can transcription and translation occur together in a eukaryote? Remember location here. Where is the DNA and where are the ribosomes? In a eukaryotic cell, the DNA, of course, is in the nucleus, and ribosomes are located in the cytoplasm. So it is not possible for transcription and translation to occur together in a eukaryote because there's a separation here. The nucleus sequesters the two activities. Okay? So transcription in a eukaryote happens in the nucleus. Translation happens in the cytoplasm. So in a eukaryote, transcription happens in the nucleus, translation happens in the cytoplasm. For a prokaryote, both transcription and translation happen in the cytoplasm. Therefore, the two can happen together in a proc, but cannot happen together in a euc because of this difference in areas. The other reason that things are more complex in a eukaryote is because a phenomenon that we call introns and exons. It turns out when we sequence eukaryotic DNA, we find that there are large pieces of the DNA that don't seem to encode anything. In fact, we call those non-coding sections introns. So introns are non-coding sections of the DNA. Exons are the coding sections of the DNA. So if we think back to how this process occurs, here we have the DNA template, complementary, antiparallel. Right? There are exons that are coding and there are introns that are non-coding, and you see introns periodically between the exons. During transcription, you have a direct copy of that DNA into RNA. Introns, exons, everything gets copied. Well, you have to get rid of the introns because they're non-coding. So eukaryotes have to splice, is what they call it. They splice out the introns and stick the exons together. That all happens before the mRNA leaves the nucleus. So there's this extra processing step that happens in eukaryotes that does not happen in prokaryotes. Eukaryotes have to splice out their introns and attach their exons together to form the mRNA. Then the mRNA can leave the nucleus. It's an extra step. That doesn't happen in procs. Procs do not have introns. Everything in a proc is an exon. Everything's coding. They don't have these non-coding sections of the DNA. So the evolutionary question here, of course, is why in the world would eukaryotes have non-coding sections of DNA? It seems like this is a big waste of time and energy to cut out these non-coding sections and all of this nonsense. Why in the world would we have evolved such a process? Short answer is we don't know, but we do have some ideas. When we talk about immunology, I'll bring this up again, and that's one reason that we know these are useful, but probably we don't know all of the reasons that we have introns. There's probably more variety than we're aware of right now, but I will tell you more about one benefit to introns when we get to immunology. All right, so differences between procs and eucs. In procs, transcription and translation can occur together, allowing them to respond to their environment very quickly. In eucs, they cannot do that because it's a location difference for transcription and translation, and also eucs have to cut out these introns or splice out these introns and put their exons together before they can go through translation. All right, so DNA mutations. So we've talked about DNA replication, structure, and how that DNA is used for transcription and translation. I want to mention why mutations are so detrimental here. First off, what is a mutation? A mutation is a change in the genetic material, so it's a change in the sequence of the DNA. So where once you had a G, you now have a C, right? That's a mutation. Now, spontaneous mutations occur constantly in all organisms. All organisms on the planet are constantly getting these DNA mutations. They occur normally at a low rate in all organisms. There are things that can increase the frequency of mutations. We call those things mutagens. So mutagens drastically increase the frequency of mutations. Things that we know that can act as mutagens, there are various chemicals that can act as mutagens. So for example, asbestos, we know various different chemicals such as acrylamide can act as mutagens. UV light can also act as a mutagen. It can have caused DNA mutations, and so can radiation. So again, mutations are normal. They occur spontaneously in all organisms, but there are occasionally chemicals that can increase the frequency of mutations. Now, why are these mutations occurring? Why do we see them occurring at all? I mean, if replication happens the way it's supposed to, you should never see a mutation. Turns out most mutations come down to a problem with DNA polymerase III. During the copying of the DNA polymerase III every now and then makes a mistake. He should have put in a T, but instead he put in a C. Right? Every now and then he makes a mistake, and so that's where most mutations originally start from, is a problem with DNA polymerase III. Now, why are mutations so detrimental to the cell? Right? DNA mutations affect, it's not really the DNA that's the problem, it's the protein that is encoded by that DNA. Remember, the DNA is used for transcription and then translation. So if you change the nucleotide sequence of your DNA, you could affect the protein that's encoded by that DNA. And that's really the problem with mutations. This is a little chart that's showing different kinds of mutations and how they could affect the resulting protein. There are really two main types of mutations, what are called substitution mutations and then frame shift mutations. A substitution mutation is when you replace one nitrogenous base with another. Right? One nitrogenous base is wrong. For example, it should have been a G and it got changed into an A. That's a substitution mutation. Now, if you change the nucleotide in the DNA, of course, you're going to change what's in the RNA, because RNA is made as a copy from the DNA. So if you've changed what's in the DNA, you've changed what's in the RNA, and if you change what's in the RNA, you change the codon. And that means you could encode a different amino acid during translation. For example, here they're showing the wild type or normal version. They're showing the wild type or normal version of a sentence here. The big bad dog ate the fat red cat. I'm sorry, just one second. Michael!
Yes.
Are you taking the kid with you?
No.
Is the oven on?
No.
Okay.
You want to, I can turn it on.
No. All right. I'm sorry for that. So the original sentence here, the big bad dog ate the fat red cat, right? That would be the wild type or normal sentence. If we do a substitution mutation where we change just one nucleotide, in this case a letter, and that's going to change the word or the amino acid that's encoded. Right? So, for example, instead of it reading the big bad dog ate the fat red cat, now we have the big bad dog ate the fit red cat. One change in a letter or a nucleotide encodes a totally different word. Right? That's a substitution mutation. Now, there's two possibilities with these substitution mutations, what are called missense and nonsense. Missense mutations encode a different amino acid. That's what we see here. The other possibility, though, is a nonsense substitution mutation. A nonsense substitution mutation means that you've changed one nucleotide and you now created a stop codon. Remember, stop codons will end translation. Now you only have half a sentence, the big bad, right? So that's the difference here. And it all comes down to one change in the nucleic acid and one substitution mutation. The other possibility here is what's called a frame shift mutation. And a frame shift mutation, you are inserting or deleting nucleotides. So you put in an extra one or you delete one, right? So an insertion or deletion mutation. Now remember, if you insert or delete nucleotides, not only are you affecting one amino acid or word, you're affecting every single codon after that. So for example, an insertion here, they put in an extra B where there shouldn't have been one. So now, instead of the big bad dog ate the fat red cat, you have the big bad dog nothing makes sense now, right? Because everything's read as 3 letters at a time. If you insert something, nothing makes sense. Same thing is true if you delete one. If you delete one, every codon after that deletion is wrong. So generally, frame shift mutations are more detrimental to the protein's function. If you change every codon after that mutation, that's going to drastically affect the shape and therefore the function of the protein. Substitution mutations, now there's a little more variety here. Sometimes there's missense ones where you just change one amino acid. Perhaps that protein could still function correctly with only one amino acid incorrect. Right? Nonsense mutations, how bad that is depends on where the mutation occurred, at the beginning of the protein or at the end of the protein. There is another type called a silent mutation. A silent mutation is a type of substitution mutation. Remember the redundancy in the code. Sometimes you change one nucleotide, but you still get the same amino acid. That's a silent substitution mutation where you have changed the DNA, but you don't change the protein because of that redundancy. So again, mutations are occurring constantly at low rates, largely because of DNA polymerase III. How bad they are depends on what kind of mutation it was, substitution or frame shift. Now, the good news is, although we're always getting these mutations, we do have a way to get rid of these mutations. This is called excision repair. This is a way to repair mutated DNA. We do have enzymes that are called excision repair enzymes, and excision repair enzymes find mutations in the DNA. Notice these two do not base pair correctly, these two do not base pair correctly. This is a mutation in the DNA where they have been mutated. Well, this excision repair enzyme is cutting out the mutated DNA, and then a different enzyme comes in and fills in with DNA, one we've actually already talked about. DNA polymerase I will fill in with the new DNA. And then ligase will seal up the gaps. So excision repair uses excision repair enzymes, DNA polymerase I, and ligase to repair these mistakes. So, I mean, it's good news that we have these excision repair enzymes. Since mutations are always occurring, we do have a way to fix those mutations. In fact, some diseases result from a lack of excision repair enzymes. For example, xeroderma pigmentosa. Xeroderma pigmentosa is a genetic disease where patients do not have an excision repair enzyme that removes mutated DNA, and for that reason they are very, very sensitive to sunlight. One of the things that causes mutations is UV light. So if the patient has exposure to sunlight and they get these little mutations, they don't have the excision repair enzymes to repair those mutations. You may have seen these sorts of cases. I've seen them on like 2020 and Dateline, where they'll show kids that they cannot go outside. They can never go outside. They can never be exposed to sunlight because they have no way to repair these mutations that commonly occur, especially in UV light. All right. The final topic that I want to talk about in this lecture is regulation of protein synthesis. And this is being regulated at the transcriptional level, so a way of regulating transcription by the cell. By the way, if you're feeling a little bit tired or sleepy, the stuff I'm going to talk about in the next couple of minutes is pretty intense and difficult, so if you're feeling that way, pause it, go eat your Wheaties, go caffeine up, whatever it is, stretch your legs, and come back to listen to it, because it's going to get a little bit intense here for the next couple of minutes. All right, prokaryotic operons. Prokaryotic operons here, an operon is a coordinated set of genes, all of which are regulated as a single unit. Now, operons only exist in prokaryotes. Highlight, underline. Only prokes have operons. And again, an operon is a coordinated set of genes, all of which are regulated as a single unit. For example, if you're a bacteria that wants to digest maltose, right? Maltose is a sugar. It doesn't take just one enzyme to digest maltose. It takes several enzymes to digest maltose. If you want to digest maltose, you need to make all of those enzymes to be able to digest it. Making only one will not help you. It's kind of an all or none thing. If you want to digest maltose, you got to make all of those enzymes. So what the prokaryote does is it puts the genes for all of those enzymes that are used together under one promoter. Right? It's a coordinated set of genes, the genes that are all involved in the same process, and it regulates them together. One promoter is going to control all of those genes so that they're either all transcribed or none of them are transcribed. Okay, so they're transcribed and then translated together. That's an operon. All right. There are two different kinds of operons, what are called inducible and repressible operons, and we'll look at an example of each. An inducible operon, in that case, transcription from the operon is turned on by the substrate. So inducible operons are normally off, but they get turned on when their substrate shows up. A repressible operon is the opposite. In a repressible operon, transcription from the operon is normally on, but it can get turned off by the product. Let's look at an example here. The quintessential example of an operon, you're going to hear about this operon over and over again. You definitely want to make sure you understand the lac operon. He shows up on all the standardized tests. This is always the one that they use as an example. All right, the lac operon. First couple of notes before we get into how it all works, the parts here. The lac operon has genes that have the enzymes needed to break down or catabolize lactose. Lactose is a sugar. So the lac operon here contains structural genes needed to catabolize or digest lactose, a sugar. Normally, transcription is off. Why? Why are these genes not normally transcribed? Because the cell is saying to itself, why should I make enzymes to digest lactose if there's no lactose in the media? Why bother making those enzymes unless there's lactose around? Right? Makes sense. Why bother doing that? So normally transcription is off, but what will turn transcription on is lactose. If lactose shows up in the media, now transcription will get turned on. Right? Lac is what we call inducible. It's normally off, but it can be induced or turned on. So the lactose operon here, here's the structure, and I listed on the right-hand side there the parts of the lactose operon here. Let's go through the parts one at a time, some of which you've already learned. Here we have the promoter, right? We're looking at a sequence in the DNA. Here's the promoter. Remember, the promoter is the binding site for RNA polymerase, the enzyme that does transcription. Right? Promoter is the binding site for RNA polymerase. It's where transcription starts. Next to the promoter is the operator. The operator is a control site that regulates access to the promoter. The operator is a control site that regulates access to the promoter. In effect, it's going to turn on or off transcription. Then we see the structural genes here. The structural genes in this operon encode the enzymes needed to digest lactose. Now, elsewhere in the genome here is a regulatory gene. The regulatory gene, he's not in the operon, he's separate somewhere else in the DNA. And the regulatory gene encodes a protein called the repressor. You're seeing in brown here the protein. So that regulatory gene encodes this protein that's made called the repressor protein. Repressors, when they're active, bind to operators. Right? So an active repressor binds to an operator. That's what repressor proteins do. All right, so let's look at how this all works. We have our promoter, our operator, our structural genes, RNA polymerase, our enzyme, and the regulatory gene that's making our repressor protein. The other thing you need to know for all of this to kind of click together is that the lac repressor is synthesized in its active form. So when it's made, he's already active. And what do active repressors do? Active repressors bind to operators. Now, what that does is when RNA polymerase comes in and binds the promoter, he physically cannot do transcription because the repressor is in his way. So the transcriptional default is off. Now what's going to show up in the media to change this scenario? What's going to show up for the cell to want to start to do transcription, of course, is lactose. Lactose acts as what we call the inducer. Lactose binds to the repressor and it deactivates it. Right? Inducers inactivate repressors. So lactose shows up, it binds to the repressor, inactivates it, so it falls off the operator. Now RNA polymerase can bind to the promoter and do transcription. Transcription occurs, you have mRNA, translation is done, you get enzymes, and the enzymes digest that lactose, right? Turns transcription on. This is why we call it an inducible operon. It's normally off, but it can be turned on or induced to produce those enzymes. I'm going to go back through it quickly in just a moment, but before I do, I want to talk about theory here. Why is all of this occurring? Imagine if I was to invite all of you over to my house for dinner, right? You guys all showed up and I'm busy in the kitchen and I'm doing lots of stuff, and I'm sure you would all be very nice guests and you would say, oh, Dr. Igo, what can I do to help you? And I would say, oh, set the table. So you go in and you open up my big chest of silverware and you see that there are soup spoons and teaspoons and there are dessert forks and there are salad forks and there are steak knives and there are butter knives and there's all sorts of different silverware. Would you set the table with every piece of silverware that I own, or would you turn around and say, what are we having for dinner? Right? You're not going to set out every piece of silverware. You're going to ask, what silverware do we need according to what we're eating? That's what the cell is doing. The cell is saying, why should I make enzymes to digest lactose if there's no lactose in the media? Right? It doesn't make any sense. Only wants to make those enzymes if lactose is there. So this is an efficiency model. So here, normally when there's no lactose, right, the repressor is made in its active form. Active repressors bind to operators, and that stops transcription. RNA polymerase cannot do transcription. Right? That's the normal scenario. If lactose shows up, however, it acts as an inducer. Inducer binds to the repressor protein. Repressor protein is inactivated. It falls off the DNA. Now RNA polymerase can do transcription in order to get those enzymes, and the enzymes can digest the lactose. That's the idea here. Now when you're trying to remember the lac operon and everything that's going on, you definitely want to know the parts of an operon, right? What is an operator? What is a promoter, a repressor, all of those things, an inducer. You'll also want to know when is the operon on, when is the operon off, and you'll also want to know what do the structural genes do. For me, if I know what the structural genes do, I can always go backwards and figure out when would it be on and when would it be off. Let's look at another example. This is the arginine operon. The arginine operon is different. The arginine operon contains the structural genes are the enzymes needed to synthesize or make arginine. Now this is different. Arginine is one of the 20 amino acids that all cells need in order to make proteins. All cells must have arginine because they need to make proteins. Okay? That's different. Remember, lac was digesting lactose. Here we're making arginine. For that reason, because arginine is vital to protein production, normally transcription is on from the arginine operon. Now, what's going to happen is if arginine shows up, then transcription will be turned off. In other words, why make my own arginine if I'm floating around with lots of it in the media? So the Arg operon is what we call repressible because it's normally on, but it can be turned off. All right, let's look at how this works. The parts again, many of them are the same. We have a promoter, binding site for RNA polymerase, beginning of transcription. We have an operator, this control site that controls access to the promoter. We have the structural genes, and the structural genes of Arg are the enzymes needed to make arginine. We have a regulatory gene. They're not showing it to you, but the regulatory gene would be somewhere else in the genome, and it encodes the repressor protein. Now it turns out that the Arg repressor is synthesized in its inactive form. So normally the Arg repressor is not active. Therefore, it does not bind to the operator. So therefore, RNA polymerase is doing transcription because that repressor is inactive. So we have transcription, we have translation, we're making enzymes, and the enzymes are synthesizing arginine. That's the default situation. Now what's going to show up in the media to change this environment is arginine. If arginine shows up, the cell is thinking, well, why am I spending so much time and energy making arginine if I'm swimming around in a buffet of arginine? So if arginine shows up, arginine acts as what we call a co-repressor. Co-repressors bind to and activate repressors. So arginine shows up, binds to the repressor, activates that. Active repressors do what active repressors do, bind to the operator. Now RNA polymerase cannot do transcription because it can't get by that repressor. Turns transcription off. Right? Why make my own arginine if I'm floating around in a buffet of arginine? That's the idea here. So the Arg operon, similar structure here. The different parts do the things that the parts always do, but it's a little bit different just based on what the structural genes do. So again, for the Arg operon, the structural genes synthesize or encode enzymes that synthesize arginine. Normally that's on. Normally you need to have the production of arginine, so this repressor is inactive. But if arginine shows up in high quantities, it will bind to the repressor and activate it. Arginine acts as what we call a co-repressor, and that allows that active repressor to bind to the operator and stop transcription. I also am posting a link to a video that goes through the lac repressor. So if you want to look at the lac repressor video, you're welcome to. It's in the same folder. I didn't find a video for the Arg operon, so hopefully you're feeling okay with operons on Arg. If not, by all means, send me a message that you'd like me to talk more about operons next time we meet, because I could certainly do that. All right, I will see you guys next time.